Hello and welcome to our next lesson in A Level Biology, looking at respiration and more specifically aerobic respiration. In previous lessons we've looked at glycolysis and we've looked at the link reaction. Today we're going to look at the Krebs cycle. So by the end of the lesson you should be able to recall the Krebs cycle. You should be able to explain what is happening to the different molecules as the Krebs cycle progresses. You should be able to explain what the waste products of the Krebs cycle are and where they come from, and you should be able to evaluate how the Krebs cycle is maintained and recycled. So let's start by thinking about what we've looked at previously. So previously we've looked at glycolysis. Glycolysis is where we split glucose into smaller molecules. More specifically, we're splitting glucose into two molecules known as triose phosphate. Triose phosphate. So we've got a triose phosphate here, and we've got a triose phosphate down here. In order to turn these glucose molecules into triose phosphate, we have to invest some ATP. So we're going to invest an ATP molecule here, and we're going to turn that into an ADP plus an inorganic phosphate. And we're going to do the same up here. We're going to invest an ATP to make an ADP and an inorganic phosphate. Just make sure we're still recording. So we've invested these ATPs, we've turned glucose into triose phosphate, and we're now going to turn these triose phosphates into molecules of pyruvate. So we've turned triose phosphate into pyruvate. And we've got pyruvate down here. Now, when we turn these molecules into pyruvate, this is where we get a payoff of ATP. So we've gone from investing these two ATPs to make triose phosphate, but when we turn triose phosphate into pyruvate, we're turning two ADP and two inorganic phosphates into two ATP. And we'll get the same happening up here. So we're going to have two ADP and two inorganic phosphates to form two ATP. Okay? At the same time, we're going to form some molecules of NADH. So we're going to have NAD being turned into NADH. Worst N in the world. NADH. And we're going to have the same down here. We're going to have NAD being turned into NADH. So we're having triose phosphate oxidized. NAD is reduced to form NADH, or reduced NAD. Now, pyruvate is then fed into what's known as the link reaction. And in the link reaction, we're turning pyruvate into a molecule of acetate. Acetate. Which is then turned into a molecule of acetyl coenzyme A. Acetyl coenzyme A. And we're going to have that at the bottom as well. Okay, apologies if I do keep checking if we're still recording. I've tried to make this video 2 or 3 times and it hasn't recorded each time, so I know I'm really worried that I'm going to get so far and it doesn't record again. So we're turning the pyruvate into acetate, and we're then turning the acetate into coenzyme A. To turn pyruvate into acetate, what we're going to do is we're going to decarboxylate it. So we're going to remove carbon dioxide from it. So we've got decarboxylation of pyruvate. And we're also going to oxidize the pyruvate. So we're going to have NAD being turned into NADH again. So we've got, if I remove that, sorry, we've got NAD being turned into NADH, like so. And we've got NAD being turned into NADH down here, like so. So we're getting decarboxylation and oxidation, okay? Acetate is then turned into acetyl coenzyme A by adding a molecule of coenzyme A to it. So we're going to add coenzyme A here, and we're going to add coenzyme A down here. So we've gone from having a single molecule of glucose, which is turned into two triose phosphates, which are then oxidized to form pyruvate. We've then got pyruvate being decarboxylated and oxidated to form acetate. We're then having acetate added to coenzyme A to form acetyl coenzyme A. Now, just for your reference, we are literally going through this just to remind you. This side, turning glucose into pyruvate, is known as glycolysis. Glycolysis. And turning pyruvate into acetyl coenzyme A is the link reaction. The link reaction. Okay? So for every one glucose molecule we're forming two acetyl coenzyme A's. So we can put, before we rub the board off, we can say one glucose makes two acetyl coenzyme A's. Acetyl coenzyme A. I'm going to be referring to this quite a lot because it seems to be confusion at A-level biology when we're talking about aerobic respiration. Okay? So, if you want to pause the video to take any notes, you can. Let's just refresh our screen for now. Make sure we're still recording. Yes, we are. And we can start looking at the Krebs cycle. So, the Krebs cycle happens in the matrix, okay, of the mitochondria. Am I going to fit that in? Mitochondria. Oh, look at that. It's almost like I planned it. So the Krebs cycle happens in the matrix of the mitochondria, okay? You need about half a page for your notes, so make sure you've got it. And as we did before for the Calvin cycle, we're going to be drawing a big cycle and talk about what happens. Now, before we look at this as a cycle, it's important that you understand that you need to be able to put this into words as well. So I am going to go through this step by step. We're going to talk about it and draw it on the screen. However, it may be worth in your notes to annotate your cycle and say what's happening at each point, because you can be given a 9-mark question, 6-mark question saying, explain what happens in the Krebs cycle. Okay? So for ease of use, purely because it's a cycle, you can start anywhere, we're going to start with a molecule known as oxaloacetate. Oxaloacetate. And oxaloacetate is a 4-carbon molecule. It has 4 carbons. Okay? Oxaloacetate becomes a molecule known as citrate. And citrate has inside of it 6 carbons. Now, oxaloacetate becomes citrate by adding a molecule, I'm not going to use, that's the worst line ever, by adding a molecule of acetyl coenzyme A. So our acetyl coenzyme A is fed into the Krebs cycle here. So the acetyl coenzyme A has 2 carbons in it, and is fed into the Krebs cycle at this point. So we have acetyl coenzyme A being added to oxaloacetate to make citrate. When this happens, the coenzyme A is released. So coenzyme A is released. Okay? So we've got oxaloacetate being added to acetyl coenzyme A, which has come from the link reaction to form citrate. Coenzyme A is released. 4 carbons plus 2 carbons makes a 6-carbon molecule. The citrate is then converted into a molecule known as alpha-ketoglutarate. Okay? Alpha-ketoglutarate. Now, it doesn't just become alpha-ketoglutarate, okay? Alpha-ketoglutarate is a 5-carbon molecule. It's made up of 5 carbons. Therefore, we're going from a 6-carbon molecule to a 5-carbon molecule. There is a loss of carbon dioxide here. So we get decarboxylation in this process. At the same time, the citrate is oxidized, okay? It has hydrogen removed from it, and that hydrogen, that wasn't the colour we were using, was it? That hydrogen is added to an NAD molecule to make NADH. So to turn citrate into alpha-ketoglutarate, we are having decarboxylation and oxidation. So at this point here, we've got decarboxylation and oxidation. So we've got the decarboxylation and oxidation, oxidation of citrate to form alpha-ketoglutarate. Alpha-ketoglutarate is then turned into a molecule known as succinate. S-U-C-C-I-N-A-T-E. And succinate is made of 4 carbons. It's made of 4 carbons. Okay? So we're converting alpha-ketoglutarate into succinate. We're going from a 5-carbon molecule to a 4-carbon molecule. Therefore, we are going to have decarboxylation again. So we've got the loss of a CO2 molecule from alpha-ketoglutarate. So we've got CO2 being removed. We have also got oxidation taking place. So again, we're going to have an NAD being turned into an NADH. So we've got NAD becoming NADH, and we've also got here an ADP being turned into an ATP. So we've got an ADP and an inorganic phosphate being turned into ATP. So we've got ADP being turned into ATP. So to turn alpha-ketoglutarate, a 5-carbon molecule, into succinate, a 4-carbon molecule, we've got decarboxylation and oxidation. So decarboxylation and oxidation. Help if I could spell oxidation. To form succinate. Succinate is then turned into oxaloacetate through simple oxidation. Okay? So in a process of oxidation and oxidation alone, we've got NAD becoming NADH. And we've also got another molecule, which we haven't spoken about yet, but we've got FAD or FAD becoming FADH. Now, although we haven't spoken about FAD yet, it does exactly the same role as NAD. Okay? So we've got oxidation taking place to turn succinate back into oxaloacetate. Oxidation. So if we go through this one more time, remember you need to be able to put this into words. We've got oxaloacetate being added to acetyl coenzyme A to form a 6-carbon molecule known as citrate. Coenzyme A is released in the process. Citrate is then decarboxylated and oxidized. It has carbon dioxide removed from it, and it donates a hydrogen to NAD to make a 5-carbon molecule known as alpha-ketoglutarate. Alpha-ketoglutarate is decarboxylated, carbon dioxide is removed from it, it is oxidized, it gives a hydrogen to NAD to form NADH, and ATP is produced in the process to turn it into a 4-carbon molecule known as succinate. Succinate is then oxidized by NAD to form NADH and FAD to form FADH to form oxaloacetate again. Okay? This is known as the Krebs cycle. This is the Krebs cycle. Remember, this all happens in the mitochondria. And I said when we started this, it is useful to be able to draw the Krebs cycle like this. You definitely need to be able to memorize it. But it is also expected that you're able to put this into words and explain what is happening. So the final bit of this lesson, we're just going to clear the screen. Remember, you can pause the video and go back should you need to. So we can just put the subheading, products of the Krebs cycle. The products of the Krebs cycle. And it's important to note, because this is where people often go wrong, that for every glucose you get 2 cycles of the Krebs cycle. This is because every glucose forms 2 acetyl coenzyme A's. So we can say that 1 glucose, and I'm putting it in regs, it's important, 1 glucose forms 2 acetyl coenzyme A's, acetyl coenzyme A, so there is 2 turns of the Krebs cycle for 1 glucose. For 1 glucose. Okay? Now, to talk about the products of the Krebs cycle and what happens to them, we're going to draw a table. Our table is going to look like this. In the first heading, we're going to have product of the from Krebs cycle, product from 1 Krebs cycle, extend that a bit, and the second column we're going to have where it goes. Okay, the first molecule we need to talk about is 1 coenzyme A. So for every turn of the Krebs cycle, you're going to have 1 coenzyme A released, and it's reused in the link reaction. So this is reused in the link reaction. So the coenzyme A that is released is reused in the link reaction. At the end of every Krebs cycle, we're also going to have 1 oxaloacetate. 1 oxaloacetate. So remember, although it's a cycle, if we start at oxaloacetate, we're going to end with an oxaloacetate, and this is reused, or just used, in the next Krebs cycle. We've also got 2 carbon dioxide made, 2 carbon dioxide. And this is released as a waste product. Released as a waste product. So in terms of humans or animals, this is usually exhaled during ventilation. So we get rid of carbon dioxide by exhaling it. If you're a yeast doing aerobic respiration or whatnot, it's released by just being released into the atmosphere. We also make 1 ATP. This ATP is made when alpha-ketoglutarate is turned into succinate, and the ATP is just used as energy. Now, this energy could be for any sort of process. It doesn't necessarily have to be for anything in particular. It's just left in the matrix of the mitochondria, so the mitochondria can use it or the cell can use it. And finally, we make 3 NADH and 1 FADH. Now, these are strong reducing agents. So these reducing agents are used in the electron transport chain. So we can just put that these are reducing agents because they have the ability to donate these hydrogens. So reducing agents used in the electron transport chain. For oxidative phosphorylation. So the NADH and FADH are used as reducing agents in the electron transport chain for oxidative phosphorylation. What we mean by this oxidative phosphorylation is that we're going to make ATP using oxygen. So the Krebs cycle forms one coenzyme A, one oxaloacetate, two carbon dioxides, one ATP, three NADH and one FADH. The coenzyme A is reused in the link reaction. Oxaloacetate is used in the Krebs cycle again. Carbon dioxide is released as a waste product. The ATP formed is used as energy, and the FAD and NADH are fed into the electron transport chain, where they're used to make ATP using oxygen. Now remember, this is for one Krebs cycle. For every glucose, you would have two of this. Keep that in mind when you're asked to work out the number of products formed in a reaction. So you should now be able to recall the Krebs cycle. You should be able to explain what's happening to the different molecules as the Krebs cycle progresses. You should be able to explain what the waste products of the Krebs cycle are and where they come from. And you should be able to evaluate how the Krebs cycle is maintained and recycled. If you have any questions, don't hesitate to ask me, either email me or contact me somehow, and I will get back to you as soon as I can. Thank you for listening.